Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I welcome you back in Re-NEET Bounce Back series guys. In today's session we are going to complete our class 12th Zoology. Good evening all of you, and as you can see it here guys, your Re-NEET All India mock test series is going on every Wednesday, Friday and Sunday. Is that clear? From 2 to 5 p.m. So make sure that you lock in, you put alarms at this time for 1:30 so that you can get yourself prepared for the upcoming All India mock test thrice a week. So you have to practice these questions. Good evening all of you. Yes guys, good evening. Ma'am, upload the notes. Yes, Naveen, I'll do that. I'll do the needful today because I'm going to complete the notes. So actually, they were all in one PDF, so I thought I'll update them together. I'll do that today. I'll do that. Good evening, all of you guys. Quickly like, subscribe, share as you know the drill, and we can start with the today's session. Hi Akshay. Good evening, guys. So shall we start? Are we all ready? Give me a thumbs up that you're all ready with your notes or your NCERTs, and we are going to revise these chapters today. So guys, starting with microbes in human welfare. So this whole chapter is full of ratification. We have so many things to learn like as such, right? So make sure that you make these tables along with me. Let's quickly jump onto this and quickly revise. Am I perfectly audible? Audible? Visible? Can we start now? Really, your plant physiology was excellent. Thank you, Naveen. Thank you so much. Thank you so much. All right, guys. Let's get started. I am really happy to know that we are able to help you. So that's our whole motive, and your appreciation is everything for us. Thank you. All right, guys. So starting with this chapter, so I am going to start with, first of all, microbes in household products. So which all are used in household? So first of all, for lactic acid, bacteria is used in converting milk into curd. And which one? That is lactobacillus. And this process is known as curdling of milk. This is known as curdling of milk. And guys, by the way, milk, as compared to milk, curd is considered better. It has a better nutritional quality, more vitamin B12, which is not found in milk. And also, curd has good bacteria. So it inhibits the disease-causing microbes in the stomach. So these are the two benefits of taking curd and more than milk. Then talking about, so basically what is lactobacillus doing? So all these good bacteria and good microorganisms that we are going to study in this chapter, they are sources of some or the other enzymes. For example, this lactobacillus, what does it do? So milk has the sugar called, milk sugar is known as lactose, right? This milk sugar is converted into, that lactose is converted into lactic acid by this bacteria called lab, and lactic acid then helps in the coagulation of milk proteins. And this whole process is known as curdling of milk. Is this clear? This process is called curdling of milk. Clear? Now, moving on to the next, that is fermented foods. In fermented foods, which are the fermented foods that we are eating? It is idli, dosa, right? So these fermented products are also prepared by microbes, for example, lactobacillus or yeast, and again, good microbes. Then there is a very important term which is called toddy. Now, toddy is a traditional drink developed from fermented sap of palm. Now, guys, toddy is a South Indian traditional drink. It is a traditional drink of South India. And how is it developed? From the fermentation of the palm sap, from the sap of palm. So, of course, because it is a fermented drink, guys, that is why it will contain it will have alcoholic content. So we call it the traditional desi alcohol for South Indians. Is that clear? Sometimes we get to see, we get this name, it's called tadi. I'm sure some of you might have heard this word. We call it toddy in English, but otherwise in like South Indian peoples, I've heard it in movies, they call it taddi, and this is their alcoholic drink. Then talking about other household products, then we have, guys, just a second. Yeah. So, yes. So we have cheese also. So cheese is one of the oldest products, household products, which is produced by microbes, right? Now, depending upon which microbe are we using for producing this cheese, we have different types of cheese. For example, there is Swiss cheese. So Swiss cheese has big holes. You might have seen these Tom and Jerry, right? Eating these cheese slices. So Swiss cheese has large holes. It has large holes. So large holes are because of, so what is responsible for causing these large holes? So it is carbon dioxide evolution. So release of CO2 will produce large holes in Swiss cheese. Now we are talking about microbes, so we must know which microbe is responsible for producing the Swiss cheese. It is a bacteria, first of all. It is a bacteria named Propionibacterium shermani. Propionibacterium shermani. Is that clear? So this is the name of a bacteria. Similarly, we have one more example. This is Swiss cheese. Second example given here is Roquefort cheese. Now this Roquefort cheese is not produced by a bacteria, rather it is a fungus which can produce it, a mold, that is Penicillium roqueforti. So depending upon the microbe used, we'll have different texture and different taste, flavor of these cheese. Are these points clear? Penicillium roqueforti, right? All right, guys. Moving on to the next. So we have microbes in industrial products. So microbes used in industrial products, for example, we are going to talk about alcohol here, in alcohol and antibiotics. The two industrial products that we are going to study are alcohol and antibiotics. And first of all, the big vessels where these industrial products are produced, be it alcohol and antibiotics, the large vessels where these products are produced for commercial production. So large vessels which are used for commercial production of these products, commercial production, these vessels are known as fermenters. These large vessels are known as fermenters. Is that clear? First of all, this. Now, talking about alcohol. So the microbe that we are going to use here, is Saccharomyces cerevisiae, which is a yeast, right? And because it is used for brewing, so we also call it a brewer's yeast. Now, let's go a little back. So this same yeast, Saccharomyces, it is also used for making bread. So be it bread or be it alcohol production, both use the same yeast. So because the yeast is helping in baking as well, we call the same yeast as baker's yeast also. And just a mechanism would be different, but the microbe is absolutely same. And because it is helping in brewing, that is alcohol production, we also call it brewer's yeast. It is the same yeast which is Saccharomyces cerevisiae. Is it clear? It is a unicellular fungus. We have read about it in biological classification. Now, what is the process that undergoes? So this helps in fermentation. So the process involved is called fermentation, that is incomplete oxidation of glucose. So how do we summarize it in the form of equation? So that is C6H12O6, that is glucose, and in the presence of this Saccharomyces, it gives us ethyl alcohol plus CO2. Is it clear? Process is fermentation and it is the same yeast. Now see here, it is incomplete breakdown. So yeast that we are using here is a source of enzyme, called zymase. Is that clear? It is a source of enzyme called zymase, which helps in alcohol production as well as CO2. Now, if we talk about bread formation, it is because of the CO2, that spongy appearance occurs in bread. So this gives spongy appearance to bread. So we all know the basic process that we knead the dough with yeast added to it, and we leave it undisturbed for a while, and then we see there is swelling of the dough that swells up. We call it, that swelling up is known as leavening of dough, right? And why does that leavening or the raising up of the dough occurs? It is because of CO2. And now when you put it in the oven, the CO2 evolves even more, giving it spongy appearance, right? And this ethyl alcohol gives acidic nature to that fermented dough. And of course, alcohol is acidic. Are these points clear? Right? So that comes under household products. Now the same process occurs here also. Now, depending upon what process you are using. Now see, if I use the same process for alcohol production, now you have to understand here, during alcohol production, this yeast, we know alcohol is acidic, right? Alcohol is acidic. So this acidic content of alcohol itself is enough to kill the yeast. So maximum alcohol that you obtain, alcohol obtained by natural process or by natural fermentation, is just 13%. Natural process is just 13%. Why? Because beyond this, the acidic nature of alcohol will kill the yeast, right? Because acidic nature of alcohol kills the alcohol kills the yeast. So in case you want to obtain higher potency of alcohol, higher or hard liquors, so for that you will have to distill it. So thereby, guys, we have two types of alcohols. We have two types of alcohols based on the process involved. So you must always know the basis. Basis is the process involved. Is that clear? So what is the process that we are going to involve here? That is distillation. So there are types of alcohols which are obtained without distillation. We call them non-distilled or undistilled. So non-distilled are basically obtained by natural process, right? And those which will be obtained after distillation, we'll call them distilled alcohols, right? Now obviously, because this is distilled, so it will have higher alcohol content. Is that clear? And this has lower alcohol content. So because it has higher alcohol content, that will be, so therefore we call these distilled alcohols as hard liquors. Is that clear? Hard liquors. All right. So non-distilled alcohol, which are, so you have to remember the names. One is beer and second is wine. So maximum they have they have up to 12% of alcohol. Is that clear? 3 to 6 and then about 9 to 12%, that is in wine. So beyond this, if you want hard liquors, like we have whiskey, brandy, you have to learn these names. So which one are the examples of hard liquors or the distilled ones? Whiskey, brandy, rum, vodka, all these spirits or the absolute alcohol, that is 100% alcohol only used for laboratory purpose, they are all distilled alcohols. Are these points clear? Is this clear to all? So guys, industrial products in alcohol, so used in making all these, so this involves anaerobic fermentation, and we have seen which types are distilled and which ones are the non-distilled. Then coming on to antibiotics. So antibiotic name itself suggests that it is against life. Now the name is a sort of misnomer, but if we talk about organisms, see here, if we talk about antibiotics, then with respect to humans it is pro-life, but if we talk in concern with microbes, for microbes, with respect to microbes, it is against life, so it's going to kill the microbes. And with respect to humans, it is pro-life. So the name is with microbes. So for humans, it is pro-life. Now, these antibiotics are again produced by some of the other organisms. So these are the chemicals produced by one organism that kills or retards their growth or retards the other organism. Is that clear? So these are antibiotics. Now please see that these are produced by one organism and retards the other organism. So it is usually a bacteria. It is never a virus. Why? Because it kills another organism. Virus is not considered an organism, and moreover, if we go into the details and understand the mechanism in which they work, see, you will see the mechanism of antibiotic in which the antibiotics function. They either destroy the cell wall or they will destroy the plasma membrane, or they will destroy the small subunit of ribosomes of bacteria. So let's say 30S subunit of ribosome, or it destroys the larger unit, that is 50S, right? Or it prevents the translation. So none of these components are present in virus. So that is why antibiotics are not used against not used against viruses. So they are used to treat the bacterial diseases. So used to treat for treatment of bacterial diseases. Please tell me all these points are clear, right? So the first antibiotic which was discovered was a serendipity. That means it was a discovery by chance. And who did that? It was Alexander Fleming. See, first antibiotic, penicillin. First antibiotic was penicillin, coming from Penicillium notatum, which is a fungus or a mold basically. Who discovered it? Alexander Fleming, right? But was he actually looking for the antibiotic? No. So he was while working on Staphylococcus bacteria. So he noticed that this antibiotic is not letting the Staphylococcus grow. So while working on Staphylococcus bacteria, right? But Fleming was not able to truly understand the potential of this antibiotic, how we can use it for the human welfare. That was not done by Fleming. That was actually done by Chain and Fleury. So coming here, so Alexander Fleming discovered it, and then Chain and Fleury. So what did these two people do? Let's see. So he is credited with the discovery, and this discovery was also a chance discovery, which is known as serendipity. Clear? While these two scientists, they understood the true potential, established the true potential of antibiotic, of penicillin. And understanding the true potential, they used it for the treatment of American soldiers during World War II. American soldiers during World War II. Is that clear? So in 1945, all three of them was awarded Nobel Prize. (1945). Are we clear till here? Please let me know that these points are clear. Yes, guys? Clear? Yes, World War II. Perfect. So, after that, there was line of antibiotics discovered and still getting discovered. Then coming on to the next part, which is microbes helping or microbes producing some biochemicals, right? Bioactive molecules, some enzymes, right? So, and some acids. So, let's see here. So, for example, so these four, these are the enzymes produced, these are the acids produced. So, first of all, we have citric acid produced by Aspergillus niger, which is a fungus. Is that clear? Then, what is citric acid used for? It is a preservative and a flavoring agent. So, all your pickles and these jellies and jams, they have citric acid. It is edible. Okay. Then talking about acetic acid. This is produced by bacteria. Now, all the other three, Clostridium, Lactobacillus, they are all bacteria. Clear? So, what is acetobacter doing? It is producing vinegar or acetic acid Is it clear? So, acetic acid is also a food preservative. It is also a food preservative and a flavoring agent. Both ways. So, we add lot of vinegar in pickles, right? And it adds flavor as well. And flavoring agent. Coming on to butyric acid, this is produced by Clostridium butylicum. It is a solvent and it is useful in printing. It helps in etching and corroding the things. Then lactic acid, we know Lactobacillus, food preservative as well as for curdling of milk. Are we clear? Now guys, now the next question. Now, out of these organisms, you have read about Aspergillus niger in detail. Please let me know in the chat to which class of fungus does it belong to. Quickly let me know. To which class of fungus does it belong to? Yes, guys. Aspergillus niger. Yes, Maria. I've had some overhauling of the studio today. Most of the things are done. I've added some more lights. My AC is still not working. I'll get this thing sorted by tomorrow. Please cooperate today. Okay? I'll get it sorted tomorrow. That is why I had to delay the class by 1 hour. Otherwise, this class was scheduled for 6 p.m. But I thought I have to take class and we'll do rest of the things tomorrow. So it's very hot in here, but we have to complete this topic. So please cooperate. Okay? Thank you so much. That's very nice. So yes, my question, guys, is Aspergillus niger belongs to which class? Yes, guys. Ascomycetes, very good. It belongs to which class? Ascomycetes. So can you get an indirect question? Which class of fungus is responsible for producing citric acid? They'll not ask you the name of the organism. They're going to ask you the name of the class, right? Okay. Now moving on to some enzymes here and some bioactive molecules. So talking about the enzymes here, see here. So there are two enzymes, pectinases and proteases, again produced by Aspergillus, although this is not given in your, but it is used for clarifying the fruit juices, right? There is one missing out, I'll write down here. Another enzyme that you must know about is lipase. Now lipase actually comes from a fungus, Candida, but we can just skip that. Where are lipases used? They're used in detergents. Stains are good. why they are good, and why are they saying that there are bioactive molecules present in the detergents. These are the bioactive molecules that they are talking about. So in stains, in detergents, they are adding these enzymes. NCERT has just mentioned lipases, but actually detergents contain proteases also, because to remove oil stains, lipases are working, and to remove protein stains, we are using proteases. So actually it is both these enzymes which are added in the detergents, but NCERT has not mentioned about the proteases. So just keep this one. Is that clear? But I have just added to your knowledge that those bioactive molecules contain both these. Is this point clear? Then guys, we have these very important names here: streptokinase, cyclosporin A, and statins. Now see, streptokinase comes from streptococcus, and this streptococcus is it is produced by the help of biotechnology, right? So we have improved this. So this is a GMO actually. This this streptococcus can produce more quantity of streptokinase. And why do we need this streptokinase? This streptokinase has an ability to dissolve the blood clots. So it can destroy the clot, basically because of fibrolytic activity. It can break down that fibrin network which is formed during the clot formation. So if the fibrin is gone, clot is dissolved, and that's what we need in heart attack patients. So streptokinase is used in heart attack patients as first aid, heart attack patients. All those who are suffering from myocardial infarction. So the first aid that is given, the first injection that is given to heart attack patients is that of streptokinase. Second, guys, we have cyclosporin A. Now, cyclosporin A is produced by Trichoderma polysporum. Okay. Now, cyclosporin A is an immunosuppressant drug. So, it is given to those patients who have undergone any kind of organ transplant. Because whenever there is an organ transplant, your body is going to reject that, graft rejection, right? You remember, we are going to study about the graft rejection also today. So the body is going to reject that, your TC cells, cytotoxic T lymphocytes. So cyclosporin is going to suppress, make your immune system sleep down, okay, so that it doesn't know that there is a foreign organ inside your body. So Trichoderma, which is a fungus, by the way, produces it. This is a Streptococcus is a bacteria. Is that clear? Now, statins are produced by Monascus purpureus, and it is a cholesterol lowering agent. It lowers the cholesterol, so it is given to high blood pressure patients. Now there is a point in NCERT about these statins, that how do they lower the blood cholesterol level, right? So the point is that these statins work as competitive inhibitors. So they competitively inhibit the enzyme which is required for cholesterol synthesis. Are these points clear? Please tell me these points are clear to all of you. Yes, please. Done? So these points should be noted. Shall we move on now? This slide is one very important slide, guys, in the whole chapter. This slide is very, very important. I even went through laptop screenshots to find if things lost or transferring. What happened, Smitha? Is that clear? All right. Moving on to the next, we have microbes in sewage treatment. Very, very important topic. Now see, guys, first of all, what is sewage? Sewage is a municipality waste. It is a municipality waste, right? So municipality waste has your household waste, human excreta, urine, right? All these things. And also the cans and the polythenes that we have thrown in in our garbage and in the drains, right? So please understand that municipality waste has two types of components. One is the non-biodegradable waste, and it has the organic or the biodegradable waste. And both these wastes have to be treated differently, right? So the first and the foremost step is that you have to separate these two wastes. Now, if we talk about the non-biodegradable waste, this cannot be degraded by the microbes. That's why non-biodegradable. So what do we have to do with that? So either we have to reuse it, recycle it, right? Or we send for landfills. Send for landfills. Is that clear? So non-biodegradable seeks this destiny. Now, we are more concerned about biodegradable waste because we can do something about it, right? Okay. Now, first step in the sewage treatment plant, please understand, is to separate them and treat them differently. Okay, we'll see how we are going to treat each of them. Now, sewage treatment plants, they were set by Government of India and the Ministry of Environment and Forestry. Okay, so they decided that in all the big cities, the big rivers, the major rivers which are passing through these large cities and getting lot of waste, collecting lot of municipality waste. So all these rivers should have these STPs so that this municipality waste can be treated then and there before it accumulates to such an extent, right? So this Government of India has set up some plans like Ganga Action Plan, Yamuna Action Plan. So all these action plans have been executed to save our rivers, and similarly Brahmaputra, Satluj and so on, right? So we have action plans for all the major rivers passing through big cities. So if a city is too big, like large enough like Delhi, so there'll be more than one STP also. Are we clear? Okay. So what are we going to do here? Let's see. So first of all, guys, sewage, we are going to make a slurry of it. We are going to make very thin slurry of sewage. Is this clear? So slurry of sewage is now going to pass or undergo the first treatment. Sorry, it is going to undergo the first treatment where we are going to separate the non-biodegradable waste from biodegradable waste. Yes. Now see here. Point is that this non-biodegradable waste will not convert into slurry. Think of glass jars, glasses, polythenes, boulders, canes, right? All the cold drink canes that we have thrown in the garbage. What will happen to them? Nothing. They will not turn into slurry. They will not turn into thin slurry, right? So they'll be all in the solid form. They'll be all in the solid form. So we have this term sludge. We use the term sludge for solid content. Is that clear? And all this biodegradable waste, which will be in the form of thin slurry, in the liquid form, we'll call it effluent. So one which flows is effluent. Is that clear? How have we managed to separate them? By two main processes. We have passed them through filters, so filtration, and we have allowed sedimentation. So these are the two methods which have been involved to separate these two portions. Is that clear? Now, these two processes, they together form your primary treatment or physical treatment. Also, we call it mechanical treatment. Is that clear? But NCERT is using just these two terms, so let's concentrate. Otherwise, it is also known as mechanical. Is that clear? So, what are the two processes involved in primary treatment? Filtration and sedimentation. So, just imagine from one side, there's a lot of sewage coming, and in the passage we have different sized filters. So things according to the size are getting separated, right? And what could not be filtered down, that has been allowed to sediment. Is that clear? Now we know what is the fate of sludge. Now because guys, this is the primary treatment, we call this as primary sludge and we call this as primary effluent. And as I said, what is the fate of these? Recycle, reuse or landfills. Send it for landfilling. Clear? Already told you. So this is an altogether different story and we're not going to deal with this. This is about microbes. So how are microbes going to help? The action of microbes will be on this part. So action of microbes is on this component, right? So now this will undergo further treatment. We are going to discuss more about this, right? Now, because this is a biodegradable waste, first understand it has organic waste, and biodegradable waste already has that bacteria and fungi which can lead to their biodegradation, their decomposition. Why do we need STPs? STPs are just going to enhance and accelerate the process, so that the same process which would naturally take, let's say, 1 year, that would occur in just few hours. So that is the role of STPs. So before the biodegradable waste goes on getting accumulated and causes fouling of the environment, so we need to break it down, and we are going to help in that breakdown, in that decomposition. So what do? This effluent is first of all passed through aeration tanks. Very important. It is passed through aeration tanks. Now the name itself suggests you what's going to happen here. In aeration tanks, guys, there is oxygen pumped in. There is oxygen pumped in. Wait. So in this aeration tanks, what's going to happen here? Oxygen is being pumped. Oxygen pumped into. Now when this effluent will enter these aeration tanks, just imagine it like let's say a washing machine. So there is a propeller beneath it. When the water enters and oxygen is being pumped, right, what will happen? The microbes which are present in the biodegradable matter, right? So microbes in the effluent, in the primary effluent, that will increase. Those microbes in the primary effluent, they show vigorous growth. They increase suddenly. Okay, vigorous growth. So which are the microbes that I'm talking about? So oxygen actually helps in vigorous growth of these microbes. Is that clear? It is because of oxygen. So what does this indicate about the nature of these microbes? That they are already aerobic. And also we will study in ecology that decomposition is an aerobic process, right? So we need to pump in more oxygen. Is that clear? So this effluent will stay in the aeration tank for few hours till the microbes increase, and now these microbes themselves will break down the organic matter. So the major time is going to be spent in this aeration tank. This is the tank where the real process takes place. Is that clear? Now, these microbes, guys, these microbes are known as flocks. These are known as flocks. Is that clear? So the microbes which increase in number and they form masses of fungi. So masses of fungal hyphae with bacteria entrapped, right, with bacteria, which is going to break down this primary effluent. These fungal masses are known as flocks. Is that clear? So because here microbes are involved, guys, that is why this treatment is called the biological treatment. It is known as the biological treatment, and it is the second step, so we call it secondary. Is that clear? Microbes are biological agents, so that is why the name is biological treatment. Please tell me that these points are clear to all. Yes? Right? Perfect. Now, after aeration, spending few hours in the aeration tank, now effluent, how do we understand? How do we get to know that the process is done and the objective has been fulfilled? So there is a technique, there is a test which helps us check it. Now we say, see, before passing into the aeration tanks, there is large amount of biodegradable waste, so large organic matter, right? So here the organic matter is high. After passing into the aeration tanks, now the organic waste has decreased. How do we get to know this? See here. So we have this test called BOD, which is biochemical oxygen demand. This is biochemical oxygen demand. Is it clear? So what is biochemical oxygen demand? So in biochemical oxygen demand, guys, the amount of oxygen, it is the amount of oxygen needed to decompose organic matter in 1 liter of water. This is the definition, the theoretical part that you must know about. Now just understand what is the process going on. See here, in order to understand that in very simple words, let's take an example. Let's say I've taken a test tube, and in each test tube there is different amount of organic matter. It has this much, and this test tube is full of organic waste. We know that decomposition is an aerobic process. Tell me very honestly, and just using our basic knowledge, if this has just this amount of organic matter, this has more matter, organic matter, and this has the maximum organic matter, which out of these 3 test tubes will take or utilize more oxygen to degrade this organic matter? Quickly let me know. Which of these is going to use the more oxygen? Yes guys, out of A, B and C, which one is going to use? Max, Jax and Haswant and Secret. Yes, you are right. It is the third one. So the whole point is, more the organic matter, more is the oxygen needed. More the oxygen needed. Yes or no? Yes. So let's say I have taken equal amount of water. I have taken equal amount of water in all of this, but rest of the component is filled with organic matter. So more the organic matter, more is the oxygen needed, and this is what BOD is. Now here we are taking it in small test tube. We are obviously not taking it liter. We are not taking 1 liter of water, but the comparison is for 1 liter of water. So we are using the terms accordingly. Is that clear? So that means more the organic matter, more is the oxygen needed, and this is what is known as BOD. Amount of oxygen needed to decompose organic matter in 1 liter of water. Is that clear? If we so we have to take equal volumes to compare the process. To compare the organic matter. Is that clear? So that means, guys, more the organic matter, more the oxygen needed, more the BOD, right? Hashtag. So can we say BOD, higher the BOD, higher, more is the pollution. More is the pollution in the water body because of organic matter. Please tell me if you are clear with this concept. Please tell me if you are clear with this concept. Right? Now let me go back. So this is our tool to measure whether the process has been done properly or not. So if BOD is higher, if the organic matter is higher, so can I say in primary effluent there will be high BOD? Can I check the BOD of solvent after it has spent few hours in the aeration tank? So what will happen after aeration tank, the effluent that comes out, guys, the effluent that comes out is sent to this. So here it has low BOD. Yes. After it comes out of the aeration tanks, it has low BOD. Are these points clear to all? Please tell me. Yes or no? Right? So it has low BOD and now it is sent to sedimentation tanks. Now it is sent to sedimentation tanks. See, my objective has been reached. My objective has been fulfilled here. So main point is this aeration tank. Are we clear? So before passing it into the aeration tank, high BOD. After that, it has low BOD. Are we clear with this point? So who decreased this BOD? It is these microbes which are called flocks. Yes or no? Please tell me these points are clear to everyone. Right? Right? So now my objective is actually fulfilled. But I cannot just release this effluent from the aeration tank directly into the water body. Why? Because during the process I have increased the number of bacteria and fungi to such an extent that if I release this effluent directly into the water body, the organic matter must have decreased significantly, but it has large number of bacteria and fungi. Now that bacteria and fungi can be parasitic, that can cause diseases, right? So before I release it into the water body, I send it towards the sedimentation tanks. Is that clear? I send it to the sedimentation tanks. So what happens in the sedimentation tanks? Let's understand. Sedimentation tanks. From sedimentation tanks, guys, what happens? This effluent is separated into, in sedimentation tanks, what happens? The effluent which comes from the aeration tanks, so aeration tanks, it is divided into two components. So first of all, we'll have flocks sediment settled down. So the flocks will settle down and they are solid, and therefore they will form the sludge. And because this solid sludge is formed as a result of secondary treatment, I'll call it secondary sludge. And then rest of the effluent is called, this is called the secondary effluent. Is it clear? Now this secondary effluent can be released into the water bodies. This is released in the water bodies. Please tell me that these points are clear to you. I have to separate the flocks, otherwise they can be disease causing. Is this point clear to everyone? Yes? Right. Now talking about the sludge, this sludge. What will I do with this sludge? Because with every batch of sewage, this flock secondary sludge will go on increasing. So I have to take care of this as well. So what do? We split it into two parts. We'll save a small part of it and we are going to put it, we call this activated sludge, right? So this small part of the flock is put here in the aeration tank during the first step of secondary treatment, activated sludge. So along with oxygen, we are adding the already formed the flocks. So activated sludge will be passed here. So now the number of flocks are going to increase tremendously and very quickly. Do we understand this point? Yes? All right, guys. And rest of the, so this activated sludge is used as inoculum. It is used as a starter, or you can say a source of infection known as inoculum in the aeration tank. Is it clear? And what about the remaining sludge? So the remaining sludge is known as, so the remaining sludge is sent to anaerobic sludge digesters. So what do these anaerobic sludge digesters have? So guys, anaerobic sludge digesters basically have methanogens. They may or may not have methanogens, but they are certainly anaerobic bacteria which are going to destroy them. Please tell me these points are clear. Are these points clear? Right? So guys, this is sewage treatment process. Clear? Now moving on to the next and the next step, which is biogas production. So in biogas production, guys, we have these methanogenic bacteria, right, called methanobacterium, methanococcus, and these are also found in rumen of cattle. It is the same one. So we use gobar, the animal dung, for making this biogas. The major component of this is gobar, or the animal dung. That is why we also call it gobar gas, right? Now the point is, this biogas, the main component of biogas is methane. Then so 50 to 70% is methane. Then we have CO2 and other gases, H2 as well as, and rest of the other gases, sorry, H2S and rest of the other gases. So methane is the main component. Is that clear? What is the process involved? It is again anaerobic digestion, anaerobic breakdown. Is it clear? You must know about this biogas plant and the labelings. So see, we are adding dung and water in the slurry form. So this is the inlet, and this is the digester where it will have the methanogens. This digester contains methanogens. So it is around 10 to 15 feet deep, made up of concrete tank, right? And so as the methanogens work through this dung and other components, so methane gas will be produced, and this gas holder will keep on raising, right? And this is an indication that the gas production is taking place. Now, while the gas is being produced, the dung is being broken down and it will turn into manure. So we can take out the manure and use it as a fertilizer. So it is called sludge. So it is used as a manure in the fields. Is it clear? Now this whole setup is generally put up in the, not generally, it is always put up in the rural areas where there is surplus availability of animal waste. Is that clear? So now remember who developed this technology. Very important. So there are two bodies which are involved in the development of this technology. So this technology was developed by India. Technology developed by, number one, we have KVIC, and second is IRI. You must know the full forms also. First is, yes guys, is this point clear? Yes, Tanuj. Yeah. I am in a class, Tanuj, right now. Call you. Ha, okay. All right, all right. So it was Khadi and Village Industries Commission. Is that clear? So it was a collaboration of these two bodies who developed this technology for developing this green fuel. And IRI, which is Indian Institute of, Indian Agricultural Research Institute. Is that clear? Please let me know that these points are clear to everyone. Good evening, good evening. Those of you who have joined me late, guys, please go through the lecture that we have. How long is the class, Venkatesh? So guys, I'll tell you what is the format of the class here. So I'm going to, I've completed almost, so no, few topics are left: biocontrol agents, fertilizers and pesticides. Other than this, I'm going to complete two chapters: evolution and health and diseases. As far as biotechnology is concerned, in next session, that is regarding the botany, I'm first going to take genetics. That will be on Wednesday. Okay, Wednesday 7 p.m. onwards, we are going to discuss genetics. It's going to be a very important class. So please understand that after genetics, I want to discuss biotechnology. Is that clear? So I'll be discussing biotechnology in the second part of botany. I know it is in zoology, but I want to go step by step. So I have put it in the botany part. Is that clear? So genetics we'll be discussing as the part 2 of botany. Sorry, biotechnology. So Wednesday 7 p.m. I'm going to discuss genetics, right, in detail. And biotechnology is all dependent on genetics. Is that clear? Clear? Perfect. Moving on now to biocontrol agents. So what are these biocontrol agents? Biocontrol. So these are those organisms which are going to control another organism. One organism controlling another organism. That is biocontrol. For example, see here. So there is this trichoderma fungus, which is a free living fungus. It is a free living fungus. So you see it in soil. So it controls the plant pathogens. So the organisms which cause diseases to the plants are controlled by trichoderma. Is that clear? It is a fungus. Then we have Bacillus thuringiensis, BT. This controls many insect larvae. So in BT cotton, by the way, BT cotton is actually a product of biotechnology. We are going to discuss that in a lot of detail. But Bacillus thuringiensis can otherwise also control insect larvae. How? By using their spores. We dissolve these sachets of spores in water and we spray. So this bacteria produce a chemical known as sporin, which destroys the insect larva. Is that clear? And these insect larvae, these are plant pests. These are plant pests. Is that clear? Then talking about ladybird beetle and dragonflies. So ladybird beetles, they eat aphids, and dragonflies, they eat mosquito larvae. Similarly, guys, we also have gambusia fish, which is put in the paddy fields. So this gambusia fish also controls the mosquito larva. So one organism controlling another organism, this is an example of biocontrol agents. Is that clear to all? Is that clear to all? Then one very important example we have here, that is of baculoviruses. We have baculoviruses. Also guys, after I complete these marathons, which will be done by the coming in this week, so we'll have classes. I'll tell you very clearly right here before I continue, before I forget to mention. So guys, what is going to be the schedule? So today we are completing zoology except biotech. Then Wednesday, Thursday and Saturday, so we'll have class 7 p.m. onwards, and we are going to complete our botany. So this is botany 1, where I'm going to discuss genetics 1 and 2, right? Then I'll be talking about biotechnology 1 and 2 along with sexual reproduction in angiosperms. And then third lecture will go to ecology, and you must know that ecology itself gets around 10 questions, right? So it is one of the most high weightage unit. Is that clear? So this is how our plan is. So now moving on to baculoviruses. Please see, baculoviruses are a special type of viruses which are designed, which are very insect specific, very target specific. So they belong to the genus, nucleopolyhedrovirus, NPV, nucleopolyhedrovirus. Is that clear? This is baculovirus. Done. Now they are very insect specific. So when be it organism or any chemical which is very, very target specific, we call it narrow spectrum. So they are narrow spectrum. Is that clear? So because they are made insect specific, therefore they have no harmful effect on other animals or humans or plants. So that is why we use these baculoviruses, guys. These are used in integrated pest management programs, IPMs, and in ecologically sensitive areas. Are we clear? Please tell me. One of the very important ones, reproduction chapter, ma'am. Please, I am not that perfect at the chapter. Danny, don't worry. Just attend the lectures consistently. You will find that chapter very easy. And in that case, you, if you are in little hurry, Danny, then you can attend the previous lectures also. I have already discussed that chapter before NEET exam 3rd May. So in case of hurry, you can watch that. If you can wait till just Thursday I'll be taking that chapter. That is why I'm not taking a gap, guys. See here, I'm not taking a gap here. So Wednesday I'm taking genetics, and right after that day I'm taking biotechnology. So I don't want a gap in between. Is that clear to all? Please tell me. Are we clear? Okay, Danny, thank you so much. Are these points clear to everyone? right? Okay, boy. Now, coming on to the last component or part of the chapter, that is biofertilizers. So instead of guys using the chemicals, we are going to use microbes to increase the soil fertility, and those microbes are called biofertilizers. It can be bacteria, blue-green algae, or or fungi, right? For example, rhizobium, it is a symbiotic bacteria. So yes, it's symbiotic bacteria. So it lives in the root nodules and helps in nitrogen fixation in root legumes. Lives in the root nodules of legumes. Is that clear? Then Azospirillum azotobacter, it is a free living bacteria, again nitrogen fixation, but in non-legumes. Then Anabaena nostoc, it is a cyanobacteria, again nitrogen fixation and does photosynthesis. We have read it in biological classification also. Why? Because they have heterocysts. So all these which are fixing nitrogen, they have an enzyme, not heterocyst, but they all have enzyme called nitrogenase, right? And then talking about mycorrhiza, we have read about the symbiotic association of fungi. Now, it enhances phosphorus absorption as well as it releases some growth factors and protects the plant from pathogens, protects against plant pathogens. Is that clear? Now, there is one important term given in here. So, fungi, which is generally involved in forming the symbiotic association, that belongs to the genus Glomus. So guys, with this we complete the first chapter of the day, that is microbes in human welfare. Is that clear? Please tell me that these points are clear. Ma'am, what about the questions? So guys, yes, I will be sharing the whole schedule of my special classes. Please follow me on the Unacademy Learners app. Okay. And I am going to start with special classes where we are going to discuss many questions, all the practice questions. So before 3rd May also, I was discussing lots of questions in the special classes, and there was repetition of around 7-8 questions from those classes. So please understand the importance of this of those question practice. We will be doing that all in special classes. After completing these marathons, yes, there will be some sessions on YT also, but my main question practice is always on the app, absolutely free. Please come and join and discuss those questions. You can also bring in your doubts and we'll be taking that as well. Is that clear? And through questions, there will be, and questions will also be unit wise, chapter wise. So we'll be discussing the topics also along with the questions. It is not just merely running through the questions. No, we are discussing questions in detail. Are we clear? Please follow. All right, guys. Now, coming on to the next chapter. Yes, guys. So, next chapter we are going to start with is health and diseases. And once again, I have to repeat, guys, that your Wednesday, Friday, and Sunday should be reserved for all India mock tests. So, this is very important from 2 to 5 p.m. Is that clear? Please tell me that these points are clear. If there is any doubt from the previous topic, please let me know. Alright, guys. Now, let's start with the next chapter. So, you will get all these notes today in the Telegram channel, and I hope you know the Telegram channel. In case some of you have not joined the Telegram channel yet. So, guys, that is Tina Ma'am Biology. Is it clear? That's it. All right. Moving on to the next chapter, that is health and diseases. All right. Coming on to the next chapter, that is health. So let's go by NCERT step by step. So what is health? It is a state of complete physical, mental and social well-being. So guys, this was the definition of health as according to WHO. According to WHO. Is it clear? But before WHO, we had some other definitions and other concepts of health as well. For example, according to Ayurveda, or we can say Charak, the father of Ayurveda, there was a concept of good humor hypothesis. Humor hypothesis. So what does this good humor hypothesis say? So first of all, humor means a fluid. So Ayurveda believed that our body is made of certain fluids, Vata, Pitta and Kapha, if you have heard of. So all these fluids, all fluids should be in perfect balance. To be in perfect balance, and if they are in perfect balance, then we are healthy. Perfect balance to be healthy. Is that clear? If there is imbalance, one particular fluid is more than the other, then it leads to diseases. So second statement that they gave is that black bile, they said black bile causes fever. Black bile causes fever. This is what they said. Because we must know that originally bile is yellowish, greenish yellow fluid. It is greenish yellow fluid. But if it turns black, then it means it is a fever. See, in old times, there were no names of every disease. So everything was called a fever. Fever happened and the person died. They were not named exactly. It later happened when we were able to discover the agents that we started naming them, right? So this is what Ayurveda said. But this good humor hypothesis, it was criticized by William Harvey. William Harvey is also known as the father of circulation. So William Harvey criticized it, and gradually there was lot of evolution about the understanding of diseases, and we understood that diseases have an agent known as pathogen. So there is an agent which is responsible. Is it clear? Eventually we found a better definition according to WHO, who said that it is a state of complete physical, mental, social well-being. Is that clear? And not merely an absence of disease or infirmity. Is this clear? So health is affected by genetic disorders, infections, lifestyle, environment and nutrition. So good health will of course depend on balanced diet, personal hygiene, regular exercise, clean surroundings, social equality and economic well-being. These are the basic things that we already know. Now talking about disease. See, what is a disease? It is any condition that interferes with the normal functioning of the body. Is that clear? So we have different types of diseases now. First of all, there is infectious diseases, right? And then we have non-infectious diseases. So what are the differences? See, infectious disease will always have an agent causing it, pathogen. So it can be communicated from one person to another, hence we also call it communicable diseases. While non-infectious don't spread from one person to another, there is no pathogen involved. No pathogen involved. Is that clear? So these are personal diseases. So it won't spread from one person to another. While they will spread because there is an disease causing agent involved, which can spread from one person to another. So they can be bacterial, viral, fungal, protozoan and helminthic, right? So these are just few names, but depending upon the causative agent, so we have different types of diseases. Is that clear? So we have these 5 different types of infectious diseases. We'll be discussing them. And then talking about the non-infectious diseases, these are not because of any pathogen. It can be cancer. It can be diabetes, which is because of a hormone deficiency. It can be because of hormone deficiency. It can be allergy. It can be autoimmune disorder. Are you getting that? It can be autoimmune disorder, or it can be vitamin deficiency or mineral deficiency. Are you getting that? There can be degenerative diseases. Degenerative diseases are because of the old age. So these diseases will not spread from one person to another. So hence non-infectious. Now let's talk about these pathogens. I've told you these pathogens are of 5 types, and hence we have different types of infectious diseases. Now what is their mode of transmission? How do they spread or transfer from one person to another? So mode of transmission is through air, or we can say droplets. So through sneezing, through coughing, right? That is droplet infection, or coughing, that is through air. Then water, then food, physical contact, and vectors. Which are the vectors? Vectors are some organisms which are bringing these disease causing organisms. For example, we know malaria is not caused by mosquito. It is caused by plasmodium. But who brings that plasmodium inside you? It is a female anopheles. So female anopheles, right? Culex. Then we have female Aedes, right? So all these, even housefly, carrier of diseases, number 1 enemy, right? Then there are so many others, trypanosoma through CC fly. So we have all these vectors. Please tell me these points are clear, right? All right, guys. So now let's take the main diseases which are given in NCERT one by one. So talking about bacterial disease first of all, and we are going to discuss about typhoid. See here. So typhoid is caused by type Salmonella typhi. I have listed down things so that it is easy for you. How does it spread? Through contaminated food and water. What are the symptoms, guys? Sustained high fever, very important, 39 to 40 degrees Celsius. Weakness, stomach pain, constipation, headache, and loss of appetite. You will be getting all these notes on my Telegram channel. These are the lists which will help you learn these things just point wise. You don't even have to open your NCERT after that. Clear? And guys, once these marathons are done, we are going to read our NCERT. I have already highlighted the important lines, and we are going to revise the whole book, those important lines together. Before NEET also we did that. After NEET also, like this, for before Re-NEET also we are going to do that. So let's quickly go through the marathons, learn the topics in detail, and then we will be revising the NCERT as well. Is that clear? Right? Now, so what is the what is the diagnostic test? So the diagnostic test of typhoid is Vidal test. It confirms typhoid. Is that clear? And now there is a very important case about typhoid, which is typhoid Mary. So she is called the classic carrier of Salmonella, famous carrier or a classic case of this of carrier of the disease. Typhoid Mary. Basically, the real name was Mary Mellon. So Mary Mellon was a cook in girls' hostel. She was a cook. But it was known that whosoever eats the food that she has cooked will get typhoid. Why? Because she was not maintaining hygienic conditions, and she was just cooking with unclean hands, and she herself had typhoid. Now see, remember, whatever enters to your body through contaminated food and water, goes into your intestine, comes out through the fecal-oral route, right? It comes out through the fecal route. So such diseases have to be taken care of. You have to maintain hygiene so much. Otherwise also you have to maintain hygiene. But the carriers, but the patients who have suffered from diseases have to maintain hygiene to such an extent. They have to clean their hands very properly, especially after going to the washroom. right? Because they are carriers. Their fecal matter contains salmonella. So if they'll not wash hands properly, they'll be spreading the disease to the rest of the members of the family. So that's what Mary Mellon did. She spread it to the whole hostel and she was given this name, Typhoid Mary. Is it clear? Now, going on to the next disease, which is pneumonia. Now, pneumonia is caused by Streptococcus pneumoniae or Haemophilus pneumoniae. And by the way, we are talking about bacterial diseases. So, how does the disease spread? So, disease spreads through droplet infection and by sharing of the glasses and utensils. Is that clear? Then, what happens during the infection? So, the alveoli becomes filled with fluid. So, the space which should be available for the air, this is all filled with fluid. And now the surface area for gaseous exchange reduces. So, surface area for gaseous exchange reduces. Is this clear? So there is difficulty in breathing, fevers, chills, because gaseous exchange is not occurring. You're not getting enough of oxygen, and when we don't have enough of oxygen, there will be weakness, there will be bluishness on the skin, right? So bluishness on skin, lips, right? Nails, they turn gray, right? So this is all because of hypoxic conditions, that is oxygen deficiency. Are these points clear? Then moving on to the protozoan diseases, guys. Protozoan diseases we are going to discuss first as amoebiosis and second will be malaria. So we are going to discuss two, amoebic dysentery, one, and second would be malaria. So I'll be discussing malaria separately. Now, let's talk about amoebic dysentery first, or also known as amoebiasis, caused by amoeba Amoeba histolytica. And we know it is a protozoan. And which type? Amoeboid protozoan, right? How does it enter our body? Through contaminated food and water. How? It is the housefly which brings it. So, it is also vector based. Now, symptoms are constipation, abdominal pain, cramps, stool with excess mucus and blood. Is that clear? So this is what happens in amoebic dysentery. Talking about malaria caused by the agent Plasmodium. So we have different species: Plasmodium vivax, Plasmodium malariae, Plasmodium falciparum, and also Plasmodium ovale. So the most dangerous out of all of them is falciparum because it is fatal. It causes cerebral cancer. Is that clear? The vector of all these is female Anopheles mosquito. Is that clear? So I'll talk about the stages in a while. But remember, the infective stage, I'll discuss the whole cycle. But the infective stage for humans, that is sporozoid stage. So which is the stage in which Plasmodium enters the human body? It enters in the form of sporozoids. Hence, this is the infective stage. Is that clear? So the life cycle I'll be discussing, and then it is during one of the stages in human body that it produces this toxic substance called hemozoin, and this hemozoin it causes malarial attack. This causes the onset of malarial attack. So when hemozoin is released, that is when we start getting the symptoms. Is that clear? Are these points clear to everyone? Yes? Are we clear, guys? Please let me know. Yes? All right. So this hemozoin results in the onset of malarial attack, causing chills and high fever and then profuse sweating. So these are the three main symptoms and it runs in a cyclic manner. So it is a female mosquito which needs blood meal for egg laying, and that is why it acts as a vector. Are these points clear to everyone? Please tell me. Right? All right, guys. Now let's quickly go through the cycle. Now remember, this plasmodium is digenetic. Plasmodium species, they are digenetic. Digenetic means it needs two hosts to complete the life cycle. So which are the two hosts that I'm going to talk about? One is a female Anopheles mosquito, and we have female Anopheles, and then we have human. Sorry. Okay. Now, whenever there are two hosts involved, there is always one. So one of them is always primary and second is other one is secondary. So here, the Anopheles is a primary host and human is a secondary host. So what is the basis of classifying them as primary or secondary? The basis is primary host will always have the sexual stages inside it. So sexual or adult stages. So that means gamete formation, fertilization, zygote formation, all those stages, that cycle will go on in the primary host, and that is why Anopheles is regarded as a primary host. While in case of human, the secondary host, we only get to see the asexual or the larval stages. Is that clear? We get to see the larval stages. Are these points clear to everyone? Please tell me. Right? So this is the nature of plasmodium. Now, I want to clarify here itself the treatment before I start with the life cycle. So treatment of malaria is quinine or chloroquine, and this quinine or chloroquine is obtained from a plant which is known as cinchona tree. It doesn't grow here in India. Cinchona tree. It's obtained from Africa. Is that clear? It's very bitter. So from the bark of the cinchona tree, that is how we get quinine, which is used for the treatment. Is that clear? Now let's talk about the life cycle. So see. So here I'm going to discuss mosquito. The life cycle which occurs in mosquito, that is female Anopheles, who happens to be the primary host, right? And here I'm going to discuss the stages which occur in human, who is the secondary host here. So first point first, we have just read, why does Anopheles bite us? Female Anopheles bite us. Male doesn't bite us because male doesn't have to lay eggs. Female bites us because she needs iron from our blood. Now, so when she is so this is the mouth. So when the female Anopheles is taken in the blood meal, so during the blood, when the blood goes in the, so this is during the blood meal, simultaneously it is also injecting the saliva into the human body. Why? Because saliva contains anophelin, an anticoagulant. So whenever the blood comes out, it starts clotting, right? And in order to maintain continuous supply of blood, it keeps on injecting that anticoagulant, which is present in the saliva, right? Now let's imagine that this particular Anopheles has that infective stage. It has this infective stage, and what is the name of infective stage? sporozoites. So these are sporozoites, and as we can see, where are sporozoites present? These are present in the salivary glands. How they reach here, we'll come to that. Okay, so through saliva, what happens here? So while it is sucking the blood, it is taking the blood and injecting the saliva along. Are we clear? Right? Now see, so when the saliva enters the blood, and along with the saliva enters sporozoites. So these are sporozoites into the blood. This is the infective stage. So through blood, guys, through blood, it enters, or they enter, these sporozoites enter our liver. Through blood, they enter the liver, where they are going to undergo lots of metamorphosis, and many larval stages will be formed. So this is liver, human liver. These are the cells of liver called hepatocytes, and these sporozoites happen to enter in these hepatocytes and undergo lots of changes. For example, it will form merozoites, cryptozoites, right, and cryptomerozoites. So these are all larval stages formed one after the other. Is that clear? So they keep on transforming from one place to another, sorry, one stage to another. So these are all larval stages. Please tell me these points are clear. Yes, guys. Are these points clear? Right? Okay. Now, these cryptozoites can either go on and reinfecting the new hepatocytes, or they can come and enter into the blood stream. They can enter into the blood stream. Is that clear? So when it enters the bloodstream again, it enters into our RBC. It infects the RBCs. So that is why, guys, during malaria, the person suffers from severe anemia. And whenever there is anemia, your spleen releases RBC, spleen also, and your spleen is a graveyard also, right? So that graveyard enlarges, your liver enlarges. So these are some clear features of malaria. Is that clear? Now, it infects the RBCs and goes through different stages. I'll clearly just write down the name of the stages because they are not really mentioned in your NCERT. So trophozoite stage, it will have signet ring stage, right? Then there will be amoeboid stage, right? And then there will be rosette stage. So there will be different stages coming up. Now see here, it is during this amoeboid stage that it releases hemozoin. This stage is going to release hemozoin. Is that clear? It is going to release hemozoin. Are these points clear? They are going to release hemozoin. Right? Okay. So hemozoin will cause a malarial attack. Clear to all? And after rosette stage, guys, there will be some cells formed. RBCs will rupture. Now at this stage, RBCs will rupture and it will form microgametocyte, and it will form a large cell called megagametocyte. These are all diploid stages, okay? These are all 2N stages. All of them are 2N stages, starting from sporozoite. Clear? Now, whenever a new female anopheles comes and sucks the blood of such a person who is suffering from malaria, she will be picking up these microgametocytes along. So, the stage which enters the anopheles gut, this is anopheles gut, alimentary canal. So what stage does it pick? It picks these gametocytes. Micro means male, gametocyte, and mega gametocyte. Is that clear? Now, inside the alimentary canal, now these stages are going to change further. They will undergo meiosis. So these are gametocytes, not gametes. So these cells, after undergoing meiosis, will form gametes. So the male gamete is more spindle shaped, female gamete is oval. So we call it megagamete. The female gamete is megagamete and male gamete is microgamete. Are these points clear? Both of them, they will fuse, that means undergo fertilization and form a zygote. Now zygote here is motile and it is known as ookinete. Is that clear? Now this ookinete goes into the alimentary wall of the alimentary canal and forms a cyst-like structure which is called sporocyst. And there are these structures formed and eventually the cyst will rupture and these sporozoids will, the cyst ruptures and releases them into the salivary gland. Are you getting that? Please tell me these points are clear. Please concentrate, guys. Please concentrate. Okay. Now in humans, guys, we are able to see the asexual cycle. So asexual cycle of Plasmodium is known as schizogony. Now part of schizogony which occurs in the liver is called pre-erythrocytic. That is before entering the erythrocytes. This is called pre-erythrocytic schizogony. And this part is called the erythrocytic schizogony. Clear? And then this region, these all stages are the sexual stages. So this in female mosquito, it shows a sexual cycle. So this region, this part of the cycle which goes on in the gut where gametes are formed, fertilization takes place, is known as gamogony. And this part where spores are formed, sporozoids, we call it sporogony. Are we clear? Please tell me that these points are clear to all. I hope you now understand why human is called the secondary host and mosquito is called the primary host. Are these points clear to everyone? Please tell me. Yes? All right, guys. So with this, malaria is done. Now moving on to the next disease, which is helminthic disease. First helminthic disease we are going to talk about is ascariasis caused by Ascaris lumbricoides or the common roundworm. Common roundworm. Is that clear? Transmission through contaminated food and water, internal bleeding, muscular pain, fever, anemia and intestinal blockage. Okay. So you should be able to compare that which one results in intestinal blockage and which one will lead to excess mucus and blood in the fecal matter. So all these, okay. And then filariasis. So filariasis, also known as elephantiasis, caused by another helminth, which is Wuchereria bancrofti or Wuchereria malai. We call both of them the filarial worm because they cause filariasis. Filarial worm. They are round worms, by the way. Both of them, they are round worms, Ascalminthes. Clear to all? Transmission is through mosquito bite. Now, which mosquito? It is a female Culex mosquito. It causes blockage in the lymphatic vessels. So, this leads to chronic inflammation and swelling of lymph and genital organs. And there is so much of swelling that the leg becomes the size of elephant, and hence the name elephantiasis. Are these clear? Moving on to fungal diseases now. In fungal diseases, guys, we are going to discuss about ringworm, and ringworm is caused by 3 different pathogens. We have Microsporum, Trichophyton and Epidermophyton. So these are the pathogens. So they infect skin, nails and scalp. Symptoms are dry, scaly lesions and intense itching. Is that clear? And it is always favored by, every fungus is always favored by high heat and moisture. So the upcoming weather will favor these. Please tell me these points are clear. Is that clear? And then we are left with viral diseases. So in viral diseases, guys, first of all, we have common cold, then dengue and chikungunya, and then we have AIDS. We're going to discuss AIDS. So AIDS, I'll discuss separately. It is caused by HIV. Dengue is caused by dengue virus. Chikungunya is caused by chikungunya virus. And both of these viruses, they are transferred in our body by Aedes mosquito, the tiger mosquito. It bites during the day. Right? And then we have common cold. Common cold is called also known as rhinitis, right? Caused due to the rhinovirus. It is a retrovirus. Just like HIV, it is a RNA containing virus, retrovirus. And what are the symptoms? Excessive mucus flow, your nasal epithelium gets inflamed, right? So we know all those symptoms. Are we clear? Now, moving on to AIDS. So see, what happens during AIDS? So we know macrophage loves eating microorganisms, right? So whenever there is HIV and it enters our body, so like any other pathogen, macrophage is going to eat that away, like it will phagocytose. But HIV is different. So first let's discuss about HIV. So HIV has two single-stranded RNA molecules, identical, and therefore they will not become double-stranded. And they also have two molecules of reverse transcriptase enzyme. Clear? So on the envelope, it has some glycoproteins. We call these glycoproteins as GP120. You can skip that, but these are the, it has two coverings, two envelopes, and it has this GP protein present on the envelope, right? Now, why I have mentioned this? Because we know that HIV infects only those cells which have CD4 receptors, which have CD4 and CD8 receptors, right? So basically, macrophage needs to have this CD4 or CD8 receptor, and this glycoprotein will go and bind with it, just like lock and key. Now you understand why we need CD4 receptors, and these CD4 and CD8 receptors are present on macrophages, on lymphocytes. So these are the two most affected cells. Is that clear? Now, like any other virus, this HIV also is going to inject, injects the viral RNA into the host cell. Is that clear? So when this viral RNA enters, you must know, see, this is macrophage, and this is macrophage. So this is our DNA, this is the host DNA, right? This is the host DNA. Now RNA cannot bind with our DNA, right? So what happens when this viral DNA enters? It also brings its own reverse transcriptase and RT. I'll call it RT, right? Reverse transcriptase. Using its own reverse transcriptase, it is going to convert. It converts viral RNA into viral DNA. Now, once this DNA is formed, this DNA is incorporated into the host DNA. This viral DNA incorporates in the host DNA. Is that clear? Now, when this DNA is attached to the host DNA, let's say this is a viral DNA, it will become, so this is the viral DNA, it will become recombinant DNA, rDNA, right? Now, this rDNA on incorporating into the host DNA, it silences the host DNA. Now, it will use the host machinery, it will overtake the host machinery to make viral particles. Is that clear? So now this macrophage is not going to serve the purpose for which it was born. It is going to become a factory house of HIV. So it will produce more HIV particles. So what will happen? So more viral particles produced. Is that clear? So these are new viral particles produced by macrophage, and therefore macrophage becomes the factory house. Please tell me that these points are clear. Becomes the factory house. Yes, please. It becomes the factory house. Is that clear to all? Right? Shalini, if you give your full attempt, attention now, so what is the how you can get marks? So first, please let me know, are you getting these 229 out of 720 or 360? First, I need to be clear about that because the strategy differs. Yes, Shalini, let me know. All right, guys. Now moving on to the next, let's talk about immunity. Let's talk about immunity. So immunity is the ability of ability of the body. It is the ability of the body to resist the disease-causing organisms. This is immunity, right? So for that, guys, we have different types of immunity. So immunity works at different levels. So immunity works at different levels. Yes, this is the types of immunity. First, let's understand this. So we have innate immunity, right, which is present since birth. So we also call innate immunity as congenital or inborn or even genetic, because it is present since birth. So the characteristics are, it is non-specific and forms the first line of defense. And then there are 4 barriers under this. We have physical barriers, that is skin. Then mucus coating of the epithelium, so mucus, right? And also third is cilia, which pushes this mucus outside. And the fourth one is hairs in the nostril, hair in nostrils. Rest of the hair on the body are vestigial. So these are the 4 physical barriers. Then about the physiological barriers. So HCl in stomach, saliva in mouth, tears in eyes, also ear wax and lactic acid in vagina. So all these are the physiological barriers, also known as chemical barriers, right? Now all these barriers, they are not letting the pathogen to enter. So these are going to prevent the entry of pathogen. They prevent the entry of pathogen. Is that clear? In case the pathogens enter, then somehow we have cellular barriers, right? And we have cytokinin barriers. Now these cytokinin barriers are particularly against virus. They protect against virus. We call these cytokines as interferons. How they are produced and how do they work, we'll talk about them. And in cellular barriers, we have monocytes, neutrophils and macrophages. All of them are phagocytic. As far as NK cells are concerned, natural killer. These are known as natural killer cells. These work particularly against the tumor cells. These are for tumor cells. So the tumorous growths which keep on happening in the body and we don't even get to know about them thanks to the NK cells or the natural killer cells. Is that clear? So they will take care of them. Talking about cytokinin barriers, they are known as interferons and they work against the viral infection. How? Let's see. So let's say if this is our tissue which is attacked by these viral particles. Let's say these are the viral particles. So when the viral particles attack this tissue, what will happen? Now this particular cell, so the cell which is attacked by virus, this is a cell which is attacked by virus. This is going to produce a special protein. It is going to produce a special protein, produces a protein known as interferons. So who produces interferons? The cell which has already been attacked. Now before dying, this cell has produced this alarm, interferon, IF. And these are gamma interferons, by the way. Is that clear? Now these interferons are going to stimulate and alarm the neighboring cells to produce antiviral proteins. Is that clear? So what do these interferons do? They stimulate the production of antiviral proteins in the neighboring cells. Is that clear? So you have to remember certain things. Interferons are produced by cells which are already attacked and they stimulate the antiviral proteins in the neighboring cells. So it means itself also it is an antiviral protein, giving information about the virus. It is also of antiviral nature. Are you getting that? So this protein, alarming protein, is called interferon. Clear to everyone? Then guys, we have acquired immunity. In acquired immunity, what are the characteristics? Acquired immunity is also known as the immune system, or we call it specific immunity. So there are certain features of this acquired immunity. Number one, it is very, very specific. It is very, very specific. It has memory. This is a very important feature. And if there would be no memory, then the same pathogen can attack us multiple times and there will be no use of these vaccines. Vaccines won't be of any help. Vaccines are purely based on this property of our immune system. Third, so our immune system is highly diversified. It has high diversity. So you give them as many new type of pathogens you can, or antigens, and it can form a specific antibody against it. This is the nature of our acquired immunity. And fourth, it can differentiate. It has the ability to differentiate or distinguish between self and non-self, between self and non-self. Please let me know that these points are clear to you. So these are the 4 important properties of our acquired immunity. 1, 2, 3 and 4. Now these properties are not present in our non-specific immunity, and they are mediated through lymphocytes. So we have 2 types of lymphocytes, T lymphocytes and B lymphocytes. Is this clear? And there are 2 types of acquired immunity based on which lymphocyte is involved. Humoral immunity. Humor word is for fluid. In fluid we have antibodies. So it is also known as antibody mediated immunity or immune system. So B lymphocytes produce these antibodies. So responsible for AIMS or HIS, humoral immunity. And T lymphocytes, there are different types of T lymphocytes which work and provide us cell-mediated immunity, that is CMIS. Please tell me that these points are clear. Yes? Are we clear here? Right? Then talking about antigens and antibody. So guys, antigen is anything that can stimulate the antibody production. So which can stimulate the immune response, that is antibody production. So whatever can stimulate the antibody production will be regarded as antigen. Is that clear? We also call it immunogen. It is abbreviated as AG. Okay, antibodies, they are produced by B lymphocytes in response to antigen. Is that clear? In response to antigen. Is that clear to all? In response to antigen, and it is known as immunoglobulin, regarded as AB. Is that clear? It is a Y-shaped structure which has 4 polypeptide chains. 4 polypeptide chains, out of which 2 are heavy and 2 are light. That is why the general formula of antibody is H2L2. Please tell me these points are clear, right? If we go with the structure, then see the structure is, this is a heavy chain, this is a heavy chain on the inner side and light chain on the outer side. Is that clear? It is a polypeptide chain. So for every polypeptide chain, guys, we have amino group on one side and CH2 group on the other side. Yes, this is the basic nature of every polypeptide chain. We have read biomolecules. So this side will be known as N-terminal, like in any polypeptide chain we do, and this is called the C-terminal. Do we see that these light chains are on the outer side and heavy chains on the inner side? Yes. Now, why do we call them light? Because they have low molecular weight. So out of these, it is divided into 2 regions. One is a variable region and there is a constant region. Similarly, variable and constant. Now, if we talk about the heavy chain, there is 1 variable and there are 3 constants. Variable constants. Is that clear? So 1 variable, 3 constants, 1 variable, 1 constant. This is the nature of heavy chain. Is it clear? Now all these 4 chains are held together by disulfide bonds. By disulfide bonds. Is that clear? Now guys, here, see carefully, here, see, here we have, on this variable side, we have antigen binding site. So this antigen binding site, guys, is called paratope. This location is called paratope. So what is paratope? This is the place where antigen will bind, antigen binding site. So how many antigen binding sites do we see in this? 2 antigen bindings per antibody molecule. Are we clear here? Yes or no? 2 antigen binding sites per antibody molecule. Now, based on these heavy sites, we have 5 different types of antibodies. D, E, M, A, G. Delta, epsilon, M for something, and then alpha, and then we have gamma. So these are the different types of heavy chains, and on that basis, guys, we have 5 different types of antibodies. Clear? Types of antibodies. So how do we remember them? In the increasing order, Dimag. D, E, M, A, G. So maximum being G. Is that clear? So you have to remember that in the increasing order. Now, what is the benefit of each? So we call it IgD, IgE, IgM, IgA, and IgG. Is that clear? So every antibody has a different role to play in our body. Is that clear to all? Okay, let's talk more about these. So first of all, IgD. This is the least and it is found on the B lymphocytes, on the surface of B lymphocytes. E, important role in allergic reactions. So M is the largest in size, pentameric in nature. So there are 5 units held together, and this one is the largest and it is produced during primary immune response. A is the secretory antibody found in milk, that is colostrum, then tears, saliva, right? That is why secretory. And then IgG, this is the smallest, and as we can see, it is the maximum. It is so small that this is the only one which can cross placenta. It can cross placenta. Produced during the secondary immune response. And this is the antibody hashtag responsible for erythroblastosis fetalis. Remember Rh incompatibility? Erythroblastosis fetalis. Is that clear? So these are the key points that you must remember about all these antibodies. Is that clear? Clear to all? All right, guys. Now, moving on to the cell-mediated. So let's see how does cell-mediated work. So cell-mediated, that is CMIS, works through T lymphocytes, and I have told you that there are different types of T lymphocytes. We have TK, killer or cytotoxic, T helper cells, T suppressor cells, and T memory cells. So these T killer cells are responsible for graft rejection. That means any foreign organ will not be accepted, organ transplant will be rejected. This is called graft rejection. This is particularly by T cytotoxic or T killer cells, and they provide immunity against intracellular pathogens. So all of them, they have different ways. For example, just told you, this is responsible for graft rejection. This is the main manager of the entire immune system. So this stimulates the B cells to produce antibodies, B plasma cells, to produce antibodies. Suppressor is going to suppress, memory is going to generate the memory. Is that clear? Then guys, we have two types of immunity. One is active immunity and then we have passive immunity. What is active immunity? So when the body produces antigens on its own. So see, that is very important. What happens during this? Body produces antibodies when exposed to antigen. Is that clear? This is active immunity. Clear? Now, active immunity can further be of two types. We have natural active and artificial active. Now, whether it is natural or it is artificial, see, one thing is sure, body is going to produce antibodies on its own. But what do we understand by natural and artificial? Here, antigen enters naturally. This is based on how antigen enters. When? During clinical exposure, natural infection, or we call it clinical exposure. So you sit with someone was sneezing, you catch the disease, come back home, start sneezing, right? So during after a few days, that will go on its own. Why? Because you have developed natural active immunity. Is that clear? Then artificial active immunity. Here antigen is deliberately added to your body. Deliberately added to the body. How? 2 drops of life, polio drops. So every vaccine has an antigen, right? So deliberately means you do it by intention. So deliberately added to the body, for example, by vaccines. So you must know what type of immunity is provided by vaccines. Vaccines provide active artificial immunity. Is that clear? Then talking about passive immunity here. What about passive immunity? So in passive immunity, guys, in passive immunity, what happens? Antibodies, ready-made antibodies given to the body. Is that clear? Ready-made antibodies are given to the body. Now, again, these are also of two types. So we have natural passive and we have artificial passive. Right? Now, on what basis do we call this one as natural or artificial? The ready-made antibodies which are given to the body, how are they synthesized? So, if the ready-made antibodies are naturally synthesized, antibodies are naturally synthesized, right? For example, in colostrum. So mother's milk, that provides natural passive immunity. In artificial, these are lab synthesized. Artificial synthesized means lab synthesized antibodies are given. For example, anti-venom. You can't wait for the body to synthesize antibodies. You have to act in emergency, provide ready-made antibodies, or you get hurt and then you go for a tetanus vaccine, right? So in that case, it is not the pathogen which is getting made to enter. You're getting the ready-made antibodies, anti-toxoid. So basically, it is a tetanus anti-toxoid that you're getting, anti-serum. Is it clear? So this is about immunity. Clear? Now talking about the responses, the primary immune response is and secondary immune response. Primary is when, so it is the response, immune response when antigen or the pathogen enters the body for the first time. Body is exposed to the antigen for the first time. And this is during successive exposures. So when it, when you meet someone for the first time, that reaction, primary. When you're meeting this person for the second or the third time, you have that memory already. That is a secondary response. Is that clear? So this one is slow, low intensity, and disease occurs during this phase. But it is going to act for the long time. So it has so antibodies are produced. Antibodies produced during this time. Antibodies and memory produced during this time. It is long lasting. Right? While secondary response is rapid, strong due to the memory cells which are produced during the primary response, but it is short-lived. And because it is strong, therefore, no disease occurs. Are we getting it? Please tell me that you're clear about these two responses. Is that clear? Yes, guys. So passive immunity, I've already discussed. Okay, so this was just went out. Now talking about vaccination. So what is vaccination? It is a practice of injecting the vaccine. And what do we have in the vaccine, guys? When we are talking about vaccination, we are actually talking about the active vaccination. We are injecting the antigen to stimulate antibody production. Is that clear? And the real antibody production which occurs, so the antibody production that follows vaccination is known as immunization. So do you understand the difference between the two? First is injecting the antigen, that is vaccination, and then what will happen inside, that is antibody will develop and immunity will develop, that is called immunization. So there are these two terms given in NCERT. I hope you should be clear about it. Is that clear? So what does vaccine consist of? Vaccine consists of, see, it consists of antigenic proteins or the weakened pathogens. Is that clear? And what does it do? What does it do? It stimulates the antibody production and memory formation. Clear? For example, polio or smallpox vaccine. Clear to all? Now, these are the third generation vaccines, the most modern vaccines that we have, which are produced by genetic engineering. And here we have made two such rDNA vaccines. We have made many rDNA vaccines, but the two given in your NCERT, hepatitis B and herpes vaccine. Now the PYQ is, which is the host used for this? The host organism used for making these is yeast cell. Is that clear? Please tell me, these points are clear to all, right? And guys, then we have passive immunization also. What is passive immunization? In passive immunization, we directly give the ready-made antibodies. Injecting ready-made antibodies. Is that clear? Injecting ready-made antibodies is called passive immunization. Are we clear till here? Please let me know. And guys, now we are going to start with allergy. Quickly guys, let me know, are we clear? Is that clear to everyone? Right? All right. Now moving on to allergy, another type of disease. Allergy. What happens during allergy? So in allergy, basically, your immune response is overactive now, exaggerated response of the immune system. So we also call it as hyperactivity, or sorry, hypersensitivity. Hypersensitivity towards an antigen. So the agents which cause allergy are called allergens. It can be anything: dust, pollen, animal dander, food, cosmetics, right? It can be some fabric. So all these are allergens. Clear? What are the symptoms? Sneezing, watering eyes, running, difficulty breathing, and it can also be fatal, right? So for example, we have some diseases. For example, we have hay fever. In hay fever, we are allergic towards pollen grains. Why? Because pollen grains contain this protein called sporopollenin, and some people are allergic towards that. Hay fever. Clear? Then, in case bronchitis, asthma, bronchitis, we have read about them. In some cases, when the whole body goes in a state of shock, for example, if a person is allergic to gluten, is not able to eat wheat, and you give wheat to that person in disguised form, person might die. In that state, the whole body goes in a state of shock, which is called anaphylactic shock. So anaphylactic shock can be fatal. Why? Because here all the body cells go in a state of response. Is that clear? So which is the antibody involved? We have just seen it is IgG, and the chemicals which are responsible, one is histamine, second is actually it is histamine. Serotonin is there, but let's skip that because it's not given. So it is histamine. The main that you have to remember is histamine. Is that clear? So in order to treat that, so first you will take antihistaminic drugs, then body is in a state of shock, so you will give it adrenaline, and third you will give steroids like cortisol also, because it is a stress hormone. So body is in a state of shock, it is in a state of stress. Clear? Then guys, we have autoimmune disorders. In autoimmune disorders, the immune system attacks its own cells, so it fails to distinguish between its own cells and the foreign cells, and own cells will act as autoantigens. So own antigens will act as autoantigens. So it produces antibodies against autoantigens. For example, rheumatoid arthritis, eczema, right? Then we have this exophthalmic goiter, then myasthenia gravis. We have read about these, myasthenia gravis. So these are autoimmune diseases. Clear to everyone, right? So now guys, moving on to AIDS. So AIDS I have already discussed with you, life cycle as well, and now talking about cancer. So what happens in cancer? There is uncontrolled cell division because of certain chemicals known as carcinogens, because of some chemicals known as carcinogens. Is that clear? So what are the possible carcinogens? So we have some physical carcinogens like radiations. These are all radiations, right? In physical, we have radiations and we also have heat. Excessive exposure to heat can also be cancer causing. Then we have some chemicals, chemical carcinogens like tobacco, smoke, right? Nicotine is a potent carcinogen. And then we have biological carcinogens. In biological carcinogens, guys, we have viruses and we have proto-oncogenes. We all have these, basically they become oncogenes here. So oncogenes are cancer causing. We all have these oncogenes in the inactive form. So till they are inactive, it's a good news, and otherwise it leads to cancer. Now, types of tumor. So we have benign tumor and we have malignant tumor. What is the difference? Benign is localized, so it does not invade the neighboring or the distant tissue. So relatively less dangerous. Rest, it depends on the tissue which is infected, because in brain even the benign tumor can be life-threatening. Is that clear? Then malignant becomes invasive. It spreads to the other tissues. That spread is known as, spreads to other tissues is known as metastasis. Hence it is, this tumor is known as cancer. And this is a very serious matter. Is that clear? These are the types of tumors. Now, I told you proto-oncogenes, which are normally present in cells, under conditions they become oncogenes and leading to cancer. Then talking about cancer detection, guys. So there are different methods: biopsy, histopathology, radiography, CT scan, MRI, right? Then we have X-rays also. For breast cancer, we have mammography. Even a basic blood test can also give us some idea. Mammography. Is that clear? So we have bone marrow test, particularly for blood cancer, bone marrow test. Sorry, bone marrow test. Is that clear? So there are different techniques used for cancer detection. And what about the treatment? Treatment occurs at 4 levels, guys. It occurs at 4 levels. Surgery to remove the tumor, radiation therapy, in which they are radiated by gamma rays. And these gamma radiations are also from some isotopes, radioactive isotopes. Now, which radioactive isotope we will use, that will differ from the tissue which is affected by cancer. Is that clear? But these gamma radiations have to be very, very target specific. Otherwise, these gamma radiations are themselves cancer causing as well. Is that clear? Then we have Chemotherapy. In chemotherapy, we are using anti-cancer drugs. For example, we have vinblastine and vincristin. These drugs, these two drugs are obtained from, Sadabahar, Vinca rosea, a very commonly growing shrub, Vinca rosea, and it is used for the treatment of blood cancer. Is that clear? Of blood cancer. And fourth, immunotherapy. So you basically strengthen your own immune system to destroy these cancer cells. So see, we are using biological response modifiers known as alpha interferons. So your immune system should also support you from inside in destroying these cancer cells. So this is the cancer treatment. Now last part of this chapter, we have drugs. The first drug we are going to talk about is cannabis sativa. Now cannabis sativa produces a very potent, so they are the source of cannabinoids, and all these cannabinoids are hallucinogens. These are all hallucinogens. We also call them psychedelic drugs. Sorry. Is that clear? So, hallucinogens. So, the first hallucinogen we are going to discuss here is cannabinoids, and the source of cannabinoids is cannabis sativa. See, there are different drugs that come from cannabis: marijuana, hash, charas, ganja. Sorry. So these are the different types of drugs obtained from cannabis sativa. Now the point is, which is the part used? So the part of this plant used is either flowers, that is inflorescence, and leaves. Is that clear? Is that clear to all? We also call this as the hemp plant. Now, it acts on cannabinoid receptors, and which is going to affect your cardiovascular system. Clear to all? Now, the second hallucinogen that we must know about is LSD, that is lysergic diethylamide. Clear? It is obtained from the ergot fungus, the claviceps, again from Ascomycetes, Claviceps purpureus, and it is an Ascomycetes. Is that clear? Very potent hallucinogen, most potent hallucinogen is LSD. Clear to all? So this is first type of drugs. Second, guys, we are going to talk about stimulants. Is that clear? Stimulants, so they act as if there is energy rush. Okay, so the first stimulant that I'm going to talk about is cocaine. Cocaine is obtained from a plant, Erythroxylum coca, commonly known as coke or crack. It increases the alertness, causes euphoria and hallucinations if taken in excess, not otherwise, only in excess. Is that clear? And it acts on dopamine transporters. Is that clear to all? Other than this, guys, in stimulants we have amphetamines. Now, amphetamines are totally synthetic. They are not obtained from any plant or any fungus. They are totally synthetic, and they are misused by students and sportsmen and athletes and even truck drivers. So synthetic misused by sportsmen, students and drivers. Is that clear? Going for the long routes, so it helps them to stay awake. So dope test, if you have ever heard of, so a particular athlete or a sportsman was rejected in the dope test, so synthetic and misused by sportsmen, students and drivers, okay, and they are, a particular sportsman have been ejected from the particular tournament because of the dope, because he or she failed the dope test. It is these amphetamines, so they are used by them to enhance their performance. And then other than these, we have tea, coffee, caffeine, right? These both have caffeine, so caffeine is a stimulant. Is that clear? Then atropa, belladonna and dhatura, these are also sources of stimulants, but in excess even these causes hallucination. In excess act as hallucinogens. Is that clear? So this is about stimulants. And then we have depressants. For depressants, guys, the first type we are going to discuss is opioids. This is of depressant type, right? So it is going to slow down the body functions. So it is going to slow down. It slows down the body functions. Clear to all? Now, the first type we are going to discuss, or the only type we are going to discuss, is opioids. Where are they obtained from? They are obtained from the poppy plant. Obtained from the poppy plant known as Papaver somniferum. Papaver somniferum. So it belongs to the family. Family is Papaveraceae. Papaveraceae. Is that clear? Now, so it is Papaver somniferum, right? Now, the drugs which are obtained directly from opium is first is opium. Then opium can be further treated to form morphine and then heroin. Is that clear? So, how do we obtain morphine? So, by methylation we get morphine, and by diacetylation of morphine we get heroin. Is that clear? So these are the drugs obtained from the poppy plant. Clear? Now what part do we use? Part used of this plant is the unripened capsule. Unripened capsule is used. From the unripened capsule we obtain latex, and this latex, on getting dried or actually oxidized, when this latex is exposed to air, it gets oxidized to form opium. Is that clear? It forms opium. Clear? So this is diacetyl morphine. That is why, because it is formed by diacetylation. Clear? It is a depressant, slows down the body functions, and we already have their receptors. So receptors for opioids, opioid receptors are present on, very important, present on GIT and central nervous system. Is that clear? Is that clear to all? Right, guys? So this is all about opioids that you should know. This is about adolescents, that why they get into drug abuse. So you must know all these things. Clear? What are the effects? Weight loss, tremors, poor health, psychological anxiety, depression, violence, poor academic performance. So dependence can be psychological, emotional need, then physical dependence means body becomes dependent and it will not function without it. So this was all about health and diseases. Please let me know all these points are clear. And now guys, we are going to, yes. Now we are going to start with the third chapter, which is which is evolution. Let's have quick revision of evolution here. Yes. So talking about the origin of universe, this origin of universe is explained by Big Bang theory. And according to Big Bang theory, guys, please note down that according to new NCERT, it is 13 point, how many years? Quickly let me know. 13.8 billion years ago. Yes. So 13.8 billion years ago, we had our origin. Then huge explosion, Big Bang occurred, and this Big Bang was a thermonuclear explosion. It was a thermonuclear explosion. Is that clear? So Earth was formed 4.5 billion years ago, and the primitive Earth was very hot, no atmosphere. There was water vapors in the form of steam, methane, ammonia, and hydrogen present. Is it clear? So these are the points to be remembered. Now, the best theory that explains us the origin of origin of life, that is theory of chemical evolution given by Aspirants and Haldane. Is that clear? So this is for origin of life. Is that clear? So the main idea of this theory the main idea is that life arose from non-living matter. So we also call this theory as abiogenesis. But once life was formed, then life came from the pre-existing life. From pre-existing life. Is that clear? So first life came from the non-living matter, and then it came from the pre-existing life. So the underlying statement is abiogenesis first and biogenesis thereafter. Is that clear? So the primitive atmosphere was reducing in nature. There was no free oxygen. This has to be remembered. So primitive atmosphere was reducing, and it contained all these chemicals. Clear? Then, now this chemosynthetic theory was experimentally proved by Urey and Miller. It was explained by Urey and Stanley Miller. So what did he do? He did this experiment for around 18 days. And what are the molecules that he took? What were the molecules he took? So he took methane, ammonia, then hydrogen, and then water vapors. Water vapors taken at 800 degrees Celsius, all in a spark chamber. Very huge spark was generated, and when it was run continuously for 18 days, in the end, what did he get? The result was formation of amino acids. So he concluded that these 4 molecules can produce amino acids. So when there is unlimited number of molecules and unlimited energy sources and unlimited time, then it can give rise to many more things. So proved organic molecules can form abiotically and supported the Aspirants-Haldane theory. Is that clear? Now the first cellular forms, look at here. The first cellular forms came 2000 million years ago, and of course the life originated in water. Now single-celled organisms, they evolved into multicellular. So invertebrates, so when did invertebrates come? 500 million years before. Jawless fishes, so I've made them in the tabulate form, around 340 million years back. The fish with the stout and strong fins, right, which could come on land and go back in water. So this was a lobe fin. So coelacanths and lobe fins were the ancestors of the modern day frogs and salamanders. So it was a coelacanth. Is that clear? Then appearance of photosynthetic organisms which released oxygen, and eventually oxygen accumulated in the atmosphere. So what happened once the oxygen was accumulated? It resulted in origin of aerobic organisms, ozone layer formation took place, and which helped in decreasing the number of radiations coming to the environment. Is that clear? Then what are the evidences of evolution? Very important. We have paleontological. In paleontological, guys, we have fossils, which are the remains and impressions of organisms preserved, like all these, and they are the most authentic evolution. Hashtag most authentic evolution, most authentic evidences, sorry. So how do we determine the age of these fossils? So first of all, the deeper the fossils are, the older they are, and which are found on the upper layers, they are younger. But then we have more scientific way, which is called carbon dating, and other than carbon dating also we have potassium argon method and many other methods also for different fossils. So carbon for dating for determining the age, where we use radioactive C14 carbon. Is that clear? Now, talking about the comparative anatomy and morphological evidence, starting with homologous organs, guys. In homologous organs, the structure, basic structure is same. Basic structure is same. So origin is same, but different function. So same origin, same structure, but different function. Is that clear? For example, forelimbs of mammals. So they will have similar bones in all of them, but they will perform different functions. See, humans for all the purposes like eating, walking, not walking now, but eating and doing many other functions. So multi-purpose. For whale, it is for swimming. Bat uses it for flying, and cheetah uses it for running. Are you getting that? So same structures, humerus, radius, ulna, carpals, metacarpals, phalanges, but doing the different functions. So hence they are all homologous organs. Is that clear? Then guys, other than this, in plants we have tendrils of cucurbita, tendrils of cucurbita, and and thorns of bougainvillea. Now both are stem modifications. Both are stem modifications, but doing the different functions. What are the different functions here? This is for support and this is for protection. So different function, same structure, hence they are homologous. Is that clear? Then guys, we have analogous organs. So analogous means different origin, same function. If there is different origin, means different structure, but function would be same. Now why different structures are performing the same function? Because of same habitat. Because of same habitat, right? Similarly, why are same structures performing different functions? Due to different habitats. So when you go in a different habitat, you perform different function with the same structure. Is that clear to all? So guys, we have wings of birds, wings of butterfly. For birds, both are used for flight. Both are used for flight, but this is a modification of forelimb and this is a modification of skin. Is that clear? Other than this, what are the other examples for analogous organs? We have potato and sweet potato. That is one, right? This is stem, this is root, but both used for food storage and hence edible. Then flippers of penguin and dolphin. Right? Are you getting that? Similarly, leaf tendril and stem tendril, both used for the same function. So you just have to check the function here. So these are all analogous organs. Are we clear? Then coming on to vestigial organs. Vestigial means now they have been reduced, they have become functionless. So appendix, coccus, wisdom tooth and ear muscles. So these are all vestigial organs. So they all show evolution. Is that clear to all? Now Moving on to biochemical evidences. So we all have similarities in DNA, again, which indicate the common origin. Is that clear? So they all indicate common ancestry. Embryological evidences. So early vertebrate embryos show similarities. So this indicates common evolution. So this was said by Ernest Haeckel. And Haeckel gave a recapitulation theory, which is also known as biogenetic theory. Is that clear? Or biogenetic theory. Are we clear? Please tell me. But this was later on criticized by von Baer. He said no. Von Baer. Clear to all? Then talking about Charles Darwin. Darwin wrote his book Origin of Species, and there were many famous voyages done by Darwin in his HMS Beagle, which is the name of his ship, Her Majesty's ship, that is Beagle. So what were the Darwin's main observations? The main observations were unlimited reproductive capacity but limited resources. And limited resources, which leads to competition, that is struggle for existence, right? And this leads to variations. Now variations can be both positive and negative, means useful and non-useful. Is that clear? So it was Darwin. Now see, Darwin, his theory was based on this key concept of natural selection. So the two concepts, one is natural selection and second concept was branching descent. These were the two key concepts of Darwinism. Is that clear? Branching descent. These are the two key concepts of Darwinism. So what did natural selection say? Says that nature selects the advantageous, that means the positive or the useful variations, and eliminates the others. So the organisms showing the useful variations, they will survive, they will reproduce, and they will pass on these variations to the offspring, and this will eventually lead to formation of new species, that is speciation will occur gradually. Please tell me these points are clear. Right? Then moving on to adaptive radiation, Darwin's finches is one example. Then we also see adaptive radiation, guys, in next case is industrial melanism seen in moths. That is also an example of adaptive radiation. First of all, what is adaptive radiation? In which different species evolve from a single ancestral species. Why? Because in a given geographical area. Why? Because they move to the different habitats. That is called branching descent actually. Is that clear? So Darwin's finches were studied in the Galapagos Islands. The original ancestor, that was a seed-eating finch. Finch is a bird. So their different beaks evolved for different foods which were available at different geographical areas. It is an example of adaptive radiation. So another adaptive radiation is seen in Australian marsupials. This is the third example. So you have to learn the names. So marsupial radiation is an example of adaptive radiation. So they were all evolved in the area called Australia. That is where marsupials were present. So you have to learn these names. And then we have convergent evolution. We have convergent evolution, also known as adaptive convergence. So adaptive convergence, guys, can be studied. Different ancestral groups evolve similar adaptations when they move to the same habitat. For example, see, marsupial mole and mole. So these are the Australian marsupials, these are the placental mammals. So when they moved at the common place, they developed this. So you have to remember them in pairs. So learn these examples in pairs. Is that clear? So next, guys, we have Hardy-Weinberg principle. Very important. So Hardy-Weinberg said that allelic frequency will remain constant from generation to generation if evolution doesn't occur. Is that clear? So that means the gene pool remains constant. Gene pool remains constant. It does not change. But for this, if evolution doesn't occur, so what are the conditions in which it will not change? Random mating, large population, no mutation, no migration, no natural selection, no interference by humans or nature at all. So this Hardy-Weinberg equilibrium has been summarized as P plus Q is equal to 1, and on squaring we get this. So what does P indicate? So P is the allelic frequency of dominant allele. Allelic frequency of dominant allele. So we'll be solving some questions here, not in today's session but separately. Dominant allele, and this one is the allelic frequency of recessive allele. Is that clear? Recessive allele. And on squaring, this is the frequency of homozygous dominant individuals. This is frequency of heterozygous individuals, and this is the frequency of homozygous recessive individuals. Please tell me if these points are clear to all. Is that clear? So direct questions as well from this topic. So what are the factors which disturb this Hardy-Weinberg equilibrium? Mutation, recombination, all these 5 factors should be learned very well. Is that clear? And what is founder's effect? See, if a group of individuals establish a new population, that new population will differ from the original population genetically. So in this case, the few individuals which will give rise to the new population, these are known as founders. And obviously, because they are genetically different, it disturbs the Hardy-Weinberg equilibrium and leads to evolution. So there will be a new population. It is a case of special genetic drift. Clear to all? Now talking about the natural selection, guys. So we have 3 types. One is stabilizing. Then we have directional. And the third one is disruptive. Now see, what are we going to do here? So here, okay. See, what are the differences between this? Let's assume there are 3 types of individuals: small, medium, and large. So we have 1 intermediate and 2 extremes. So small and large are the extremes and medium is the intermediate feature. Is that clear? Now see, if nature supports the intermediate feature more, so there will be more of the intermediate feature in the population, medium sized or any intermediate feature. This is called stabilizing, also known as normalizing. In directional, nature will support one of the extreme. So do you see more of small and less of medium, and large are not present? Or it can be, it supports more of large, less of medium, but no small. So either of the 2 will be supported. Is that clear? And in disruptive, it will support both the extremes and not the intermediate. So you will get 2 peaks in here. So this is just the opposite of stabilizing, where intermediate is favored. Here both the extremes are favored simultaneously. So guys, this is natural. These are 3 different types of natural selection. So this is evolution of reptiles through different geological periods. So how do we learn that, guys? Please see. I'll make a different video on the mnemonics, how to learn this table. So please go through, please see. that early reptiles, see here, early reptiles, first of all, they form sauropsids and synapsids. Clear? So through synapsids, what were produced? So Pelycosaurus got extinct, leading to therapsids also extinct and forming mammals. Clear? And then we have sauropsids, which gave rise to turtles, lizards and snakes, right? And even this thecodonts, which got extinct, and then dinosaurs, and then mammal, these birds. So reptiles are the common ancestors. So do we see in which phase all these happened? So these are the different time scales which are given. Is that clear? I'll give you a separate mnemonic video to learn all these things and also the human evolution. Is that clear? So I have a quick mnemonic for human evolution and type of questions coming up for that. So we'll discuss it with the help of a small story, 5-minute story for learning everything about human evolution. So guys, that was it. We have completed now. And as I've told you in the beginning, so biotechnology will be discussed on Thursday, because on Wednesday, I am going to discuss genetics. So don't forget to join me on Wednesday, 7 p.m. to discuss more about genetics. Is that clear? And after we are done with all the marathons, guys, we have to read NCERT together. We have to practice so many questions in the special classes. To follow me on the Unacademy Learners app as well. So they are absolutely free classes where we will be practicing more questions. And also don't forget to take all India mock tests on Wednesday, Friday and Sunday. Bye guys.